YouTube now.
Okay. The young instructors then were very outstanding. Yeah.
Okay, so good morning, everyone. So we'll be starting in a minute. So just a few reminders again for our audience. Kindly keep your microphones on mute. We can entertain questions at the end of the lecture. So kindly type it in the chat, which you can find at the bottom of your screen. Or if we're relatively a small community, we can also direct our questions to the speaker directly. Okay, so thank you once again for joining us this morning for the PAASE webinars. So it's already 8 o'clock, so we can start. So for this morning, our speaker earned her bachelor's degree in chemistry from the University of the Philippines, Diliman, and her PhD degree in biochemistry from the University of Hawaii. She completed her postdoctoral studies in cell and molecular biology at Baylor College of Medicine. She is currently a professor and graduate programs director in the Department of Physiology and Cell Biology and the Winthrop P. Rockefeller Cancer Institute at the University of Arkansas for Medical Sciences, Little Rock, Arkansas, USA. Her research is focused on understanding the molecular mechanisms that drive women's health related pathological conditions, including breast and endometrial cancers, endometriosis, and pregnancy loss. Dr. Simmons serves or has served on many national and international scientific advisory and review panels and on the editorial boards of national and international scientific journals. She has published over 180 professional articles and book chapters and has mentored more than 60 undergraduate, graduate, and medical students, postdoctoral fellows, and junior faculty. She has the honor of being a member of the Philippine Science High School inaugural class of 1964, a recipient of the National Science Development Board Scholarship, and a Balik scientist both in 1995 and 2017. So this morning, she's going to share with us Addressing Women's Health Issues, Scientific Inquiry, and Lessons Learned. Let us all welcome Professor Rosalia Simon.
Thank you very much. It's nice to be here. It's nice to be talking to my kababayans and, really very relaxed atmosphere, I hope. And just providing some vignettes in terms of the work that I have done in the last 35 years. So I'm really sort of like aging. I'm in the age category. So this is, let's see. This is the outline for my talk. I'm going to talk a little bit about the background, what I study and why. And then I'm going to talk about women's pathological conditions, breast cancer, and also endometrial pathologies, endometrial cancer, endometriosis, and then end up with some of the lessons learned from having to work for 35 years as a head of my independent research group. So what do I study? I study women's health issues. And why do I study it? Because women comprise half of the global population. They are highly responsible for the perpetuation of the human species. Women's work matters, and so we have to keep ourselves healthy. And specifically, there are specific differences between health of men and women. And so we have to specifically study the issues that are health issues that are important to women. And so some of these women's health issues that I will talk about is breast cancer and endometrial pathologies in the context of drivers of pathology, which is obesity and diabetes. And so I'll start with my slide. How do I do this? Yeah.
You can hover on the bottom left, and I think you'll be able to see an arrow.
I see.
There we go.
Okay,
Yeah.
yes. Okay, so this slide shows you the biological and physiological rationale for differences in women's health versus men's health. And if you can look at it in the women, in men and women, they have this almost the same endocrine system. And endocrine are the glands that produce hormones and that drive homeostasis in the physiology of the human being. But the only difference is that in terms of two glands. In the male, we have the testes, which produce a specific hormone, androgens. And in women, we have ovaries, which produce two specific hormones, estrogen and progesterone. And so these differences drive the biology of male versus female, and because of this and these tissues not only produce these hormones, but they are also specific targets of these hormones. So there is a regulatory loop, so that a dysfunction of the production of the hormones could cause a dysfunction in these organs, and therefore cause some of the reproductive issues, some of the health issues that are unique to women or to men. So if you look at this cancer statistics, you can see that there are definitely differences in males, in females, as opposed as regards to the important, the leading cases in cancer. And you can see that while there are similar in their incidences of other cancers, lung, colon, and all the other kinds of cancers that affect both male and females. In males, prostate cancer is the predominant cancer, while in females, it's the breast and the uterus. And these are, of course, female reproductive tissues that are unique to the female species, and also are major cause of mortality in females as opposed to males. So what is estrogen and progesterone? So estrogen and progesterone are steroid hormones that bind to their respective receptors. And these receptors are proteins that are key regulators of transcription. And by virtue of altering the transcriptome in the epigenetic makeup, they can modify cellular function. And you can see here the steroid hormones produced by females, which is estrogen and progesterone, can go into the nucleus because they are, they're lipid soluble. They can go through the cellular membrane, go through the nucleus, make a complex with the specific receptors that are present for this ligands, the steroid hormones, and they can bind to DNA and cause and act as transcription factors that can cause the synthesis of messenger RNA and new proteins. And so then, because they are key regulator of transcription, it is necessary that their ligands with estrogen and progesterone are controlled in a specific way, as well as their specific receptors. And you can see here that there are complexity in these receptors. The estrogen receptor, for example, has two isoforms. Progesterone receptor also has two different isoforms. And there is a regulatory loop between the estrogens and progesterones, because estrogens can increase progesterone receptor levels, while progesterone can inhibit estrogen receptor levels. So this is why it's very important to have a balance in the production of these hormones, and which can cause an imbalance of which can cause dysfunction. So aberrant steroid hormone receptor signaling is cause of reproduction, many of the diseases in women. And there are many regulatory pathways by which aberrant steroid hormone receptor signaling can occur. It can occur at the level of the receptors themselves. There's many different, as I've mentioned, isoforms. There are two different isoforms for each. And depending upon the synthesis of this specific isoforms, it can alter the function of the steroid hormones. The receptors also have specific co-regulators, activators, or repressors, and then can alter their activity. These receptors can be located in the cytoplasm, outside the nucleus, and in the nucleus. And if there's more in the cytoplasm than in the nucleus, then they cannot function in terms of being transcription factors. There are alterations, steroid hormone synthesis, as well as in kinases that phosphorylate these receptors and alter their specific activities, and therefore their transcriptional activities. So here are some of the pathological conditions that are altered as a function of aberrancies in estrogen receptor and progesterone receptor. And you can see that these are all defects in women's health, infertility, pregnancy defects, breast cancer. 1 in 12 women in their lifetime will be a victim, will have affected by breast cancer. Adenomyosis is another a condition, rare, but it's still a condition in women. Leiomyoma is uterine fibroids. It's one of the most common dysfunction in women's reproductive health. 1 in 5 women will be affected by uterine fibrosis. Endometrial cancer, 1 in 40 women are affected by endometrial cancer, and endometriosis, 1 in 10 women are affected by endometriosis. So you can see how powerful it is with how estrogen receptor and progesterone receptor working through the steroid hormones can alter women's health, because all of this, when there is a dysfunction in progesterone and estrogen receptor signaling, you have all these potential pathologies occurring in women. So let's see. So while my talk will focus today is a little bit on breast cancer and uterine pathologies. And in these studies, we use mouse animal models and also patient samples to be able to compare the normal biology versus the pathology of these tissues. And we consider obesity and diabetes as inducers of this pathology, because both these conditions can cause chronic inflammation, which then causes overactivation of the immune response, much in the same way that COVID-19 can cause a chronic inflammation and thus overactivation of the immune response. So these conditions of obesity and diabetes can lead to abnormal proliferation, all this tumor suppression, abnormal tumor suppression, dysregulated function, and tissue destruction. And so what we study is how these mechanisms affect women's health. And we try to understand these mechanisms so that we can have strategies for its prevention as well as for treatment. So if we look at breast cancer, there are many challenges associated with breast cancer, the foremost of which is that it's highly heterogeneous. There is no single therapeutic option for the treatment of breast cancer. In fact, they say that breast cancer could never be really treated. It is just that it becomes quiescent, but because of certain environmental conditions, it can arise again, so that the cancer cells are never eliminated in the body. So that is a big challenge in treating breast cancer. So what do we do in terms of breast cancer? And here I show you that this is ductal carcinoma in situ. The cancer, the abnormal growth occurs just in the breast, but these tumors then, when they overgrow, they can squeeze their way out of the mammary tissue and can go through the bloodstream, and which is called the metastatic disease. And again, this is a very deadly disease. In fact, globally, every 60 seconds, a woman dies from breast cancer. So this is a major pathology and definitely affect the quality of life and of women. So what are the risk modifiers of breast cancer? This here I list some of those. Age, of course, as you grow old, you have a lot of DNA damage occurring, oxidative stress, the tissues don't function very well, and this is something that we cannot avoid because we all grow old. Gender, female, have more, of course, has increased risk for breast cancer, while only 1% of men can have breast cancer. They do suffer from breast cancer, but very low in proportion. Genetics, there are some mutations. BRCA1 and BRCA2 are genes that are mutated in breast cancer. Reproductive history is also a risk modifier of breast cancer. early menarche, late menopause, later age at first childbirth, which is a condition associated with increased estrogen exposure, can be associated with increased risk of breast cancer. Alcohol, obesity, lifestyle, and diet are modifiers of breast cancer, but these are something factors that we can control. And so what we're trying to understand is what is the interactions between genetics and lifestyle that could diminish the occurrence or the incidence of breast cancer. So I will be presenting three different stories regarding, I would call vignettes of the work that I have done in the last, summarizing the last 15 years of work in my laboratory. And I'll start with breast cancer work. This is a question where I try to answer, does maternal diet, high-fat diet influence breast cancer risk and female progeny? And in this work, we have, this is carried out by a postdoctoral fellow in my lab, Maria Teresa Montales. She is a UP graduate physician, and she came to the lab to do her postdoctoral work, learn research, and currently she's now an internal medicine physician here in Arkansas. And there's another student who worked on this project. But what we wanted to understand is to see how maternal diet can influence the progeny's health. And so basically trying to understand how maternal health matters in the health of their progeny. In these studies, we use a mouse model of breast cancer, which is the MMTV Wnt1 transgenic mice, which means that it is expressing high levels of Wnt1. And Wnt1 is a growth factor. Abnormal levels of it underlies breast cancer in human females. So this is the experimental design that Teresa did in collaboration with Omar, another graduate student in my lab. And what she did was that the mice were fed either a controlled diet or high-fat diet prior to pregnancy. And then they were mated with male mice. And then during pregnancy and throughout their lactation, they were also fed either the controlled diet, which is a low-fat normal diet, and a high-fat diet. And so during this period, the pups or the progenies are exposed to what their mother eats. And after they're weaned, which is postnatal day 21, which is comparable to adolescent in humans, then they're transferred to a diet that is normal in terms of fat content. And so we follow the formation of tumors, seeing then that if they form tumors at different rates, it means then that this is a function of their exposure to maternal diet. And let's see. So if we look at the mammary glands of the HFDO, which is the high-fat diet offspring, we could see that very early on, they have an early development of the mammary gland. So there's an abnormal increase in the development of the mammary glands, and also not only the mammary glands, but also the fat content in the breasts. And more importantly, those who are offsprings from mothers who have fed the high-fat diet have greater tumor incidence, greater incidence of tumors, as well as onset of tumors. So they develop tumors at an earlier age than those who are exposed to a normal diet through their mothers. And we saw that in the serum of these animals and this progeny, and this offspring, which have been exposed to high-fat diet, they have higher levels of insulin, so they are prone to hyperglycemia, which can lead to diabetes. They have higher levels of this inflammatory substance, tumor necrosis factor, which means that they have a more higher levels of inflammation. And in terms of their mammary gland, they have lower expression of a tumor suppressor P10, an apoptotic, anti-apoptotic pathway BCL2, and they also higher expression of this inflammatory gene IL-6. So you could see that at a very early age, when they are only exposed to maternal high-fat diet, they have, this progeny have already inflammatory as well as diabetes or a hyperglycemia, which can lead to diabetes. So exposure to the mother's diet can, early exposure to the mother's diet can definitely alter the progeny. And in collaboration with an associate, with a professor here at UAMS, Dr. Melnick, who is a chemist, we were able to do through metabolomics, LC-MS-MS, we show that this serum of this pups, which are exposed to maternal high-fat diet, already have cancer-associated metabolites in their sera. So, and this metabolites are associated, are found in higher levels in estrogen receptor negative as opposed to estrogen receptor positive tumors. So estrogen receptor negative is the more deadly type of breast cancer as opposed to estrogen receptor positive. So all this tells us that breast cancer risk is highly influenced by the maternal metabolic status. There is a transgenerational effect so that the mother can, even if the child is not exposed, could influence, directly influence breast cancer risk in their progeny, which suggests then that the developing mammary gland very early on is highly susceptible to maternal metabolic changes, and that changes in these metabolites in the progeny that are altered by the maternal metabolic status may underlie breast cancer risk. So maternal health does definitely can matters and thus definitely could affect the health of the progeny. So this is a very emerging concept that has now been considered a focus of investigation because you know, we are trying to understand, all the other scientists are trying to understand how does cancer originate and how it can be prevented. So another work that I would like to tell you is regarding our work on progesterone receptor interacting protein. Progesterone receptor, as I have mentioned, is binds to progesterone. Progesterone balances the effects of estrogen. Estrogen is the growth regulatory factor. Aberrant concentrations of it can increase abnormal growth and therefore could promote cancer. So we are interested on those proteins that alter the function of the progesterone receptor, so that then it could be a way of inhibiting the actions of estrogen. So we have been working on receptor co-regulators, and this receptor co-regulator that we have worked with and we have worked over the years is called Kruppel-like factor 9. It's a small protein, and it was actually identified in the lab by a former postdoc who was very adamant that, and thanks to her, she was really hard-headed. I was expecting a different protein to be interacting with the progesterone receptor, but she had, I was expecting a 90 kilodalton protein, and she keeps on repeating the experiments. It was a small protein, a 32 kilodalton protein. So, And it turned out that it was after studying making sure that she is getting the right results. It turned out that there is a small protein called Cooper-like factor 9 that interacts with the progesterone receptor. This protein was discovered by somebody in Japan, but it was never identified to be a progesterone receptor interacting protein. Since her discovery at that time, we published her study, and then came Mike Villarde, who joined my lab as a graduate student, and another postdoctoral fellow, John Mark Bona, who also came from UP. He came to the lab as a postdoctoral fellow, and together they did a number of studies, really, how would you say, very interesting studies that defined how KLF9 can mediate steroid hormone signaling. And so they found that KLF9 can indeed interact with the progesterone receptor and therefore make it more higher activity, and it specifically acts with not just with the progesterone receptor, but with a specific isoform of the progesterone receptor. It can negatively regulate estrogen receptor signaling, but in some contexts can also positively regulate estrogen receptor signaling. So the yin-yang, KLF9 interacting with the with the progesterone receptor to control growth, preventing and negatively affects the estrogen receptor to inhibit its growth, promotes a homeostasis that controls overgrowth of tissues. In several more studies, we found that KLF9 is also involved in subfertility, because fertility is a function of progesterone. And it also, those mice, we were able to get knockout mice for KLF9, and found that those mice which lacks KLF9 have prolonged labor. So it's involved in early pregnancy, and it's also involved in later pregnancy. And we were able to show this not only in the mice, but also in humans when we worked with another former student of mine who was a director of OBGYN up in Pennsylvania. And she was able to get patients with normal labor and also with prolonged labor, those who are over 39 weeks of pregnancy, and found that those who are having difficulty in giving birth and prolonged labor have lower expression of KLF9. So aside from finding that KLF9 mediates steroid hormone signaling, we were able to also correlate it with some infertility pregnancy problems. So we tried to understand what is happening, and we found that KLF9 is an anti-inflammatory and anti-growth factor, have anti-growth factor activity. And this is just a gene array. You don't really have to worry too much about it, but just the differences in color means that there are changes in gene expression. So if the cell lines which express KLF9, which if we remove the progesterone receptor, you could see that there is a change in expression relative to the normal uterine cells. But if we remove both KLF9 and progesterone receptor, there is definitely another change, suggesting then that KLF9 is a progesterone responsive, progesterone interacting protein, causes a change in inflammatory signaling, and therefore the loss of KLF9 plus progesterone has a greater tendency for inflammation in the reproductive organs. So let's see. So another disease or another pathology that is involved in KLF9 is endometriosis. I don't know if you guys are familiar with endometriosis. As I said, it's a very common disease, very painful, where the uterine tissues are locate outside and honed into specific sites outside of the uterus. So these uterine tissues during menstruation are discarded. They come out, and instead of being going out, they go the other side. It's called retrograde menstruation. They go back. They can hone themselves into the ovary, into the lung, into the liver, peritoneum, and so on, and causes a lot of pain. And it's not cancer, but endometriosis has been associated now with increased ovarian cancer risk. So it is a disease that is really being put a lot of focus on. And these are the people who had worked with me in the lab. And I'm not going to describe the whole studies that we have undertaken of this. Suffice it is to say is that we found that the loss of KLF9 can cause an inflammation, and inflammation then can cause an activation of a signaling pathway, a growth signaling pathway, which will cause the growth of these ectopic lesions. Note that in these studies, we built upon the studies of other investigators. In previous studies before us, it has been shown that an estrogen receptor, increase in the estrogen receptor, is associated with endometriosis, and that it can cause inflammation, and that a decrease in progesterone receptor signaling could cause also endometriosis. So building upon all this, we were able to get a regulatory pathway by which loss of KLF9 is involved in progesterone signaling, as well as in an estrogen in inflammation causing endometriosis. What is unknown right now and is the subject of many investigations is how KLF9 is lost to cause inflammation, and what other inflammatory pathways are involved to propagate the formation of these ectopic lesions. So we also found that KLF9 is involved in endometrial cancer. I had an MD PhD student, Christian Simmons, who worked on this project. And if you look at the uterus, for those who are not familiar, this is the shape of the human uterus, and these are the ovaries on both sides. And the ovaries are the ones which produce estrogens and progesterones. And endometrial cancer is when there is a growth of tissues, overgrowth of tissues inside the uterus, and this can cause. And these are tumors, and so when they can also get out of the uterus and can metastasize to other organs, and also could be very deadly. Christian found that if you look at the tumors at different stages of pathology, where the stage 3 and 4 are more pathological than this normal and stage 1, he found that there is definitely a decreased expression of KLAD as opposed to the more normal tissues. We collaborated with Gyn-Onc here at UAMS. We were able to get endometrial tumors and adjacent normal endometrium, and we found that indeed in the normal endometrium, KLF9 is expressed, but in the endometrial cancer, in the tumors, there is a decrease in KLF9. So this indicated to us that KLF9 is indeed associated with endometrial carcinoma. Okay. So the take-home message here is that KLF9 is an important contributor to steroid hormone signaling, and loss of KLF9 by some mechanism which we have not understood at the present leads to aberrant progesterone receptor signaling, resulting in abnormal cell proliferation because there is no balance to the estrogen, which causes growth. And there's a lot of immune dysfunctions, and there's an increased expression of tumor promoting genes. So KLF9 is at the top of the mechanism, top of the regulatory mechanisms that can be correlated with endometrial carcinoma. So loss of KLF9 is associated with endometrial cancer and endometriosis, and likely with ovarian cancer because endometriosis has been shown to lead to increased ovarian cancer risk. So in a third story that I'm going to tell you, we have tried to understand how we may be able to target endometrial cancer. So endometrial cancer, unlike other cancers, the incidence and mortality continues to grow. And that 50% of endometrial cancers are attributable to overweight and obesity. And so maybe that is the cause. It's continuing to grow because of the increased obesity, especially in the more developed countries. In the United States alone, I think 30% of adult women are in the overweight, obese category. And so the key players, of course, associated with overweight and obesity are productions of high levels of estrogens by the fat cells, by the adipocytes. There's a lot of inflammatory cytokines. Estrogens can trigger a lot of immune response. When there's also obesity, there's a high production of insulin, which can cause hyperinsulinemia, higher levels of glucose, and those insulin and glucose can also are growth factors, can target other organs, and therefore can cause inflammation as well as overgrowth. So we try to understand how we may be able to correlate, have some mechanisms for targeting endometrial cancer. And so we thought that metformin might be a potential therapeutic agent for that. This is not novel idea from us. A number of people have found previously that for those that metformin, which is the first line of treatment for people with diabetes, has also been correlated with a decrease in cancer, but in other cancers, not necessarily endometrial cancer. So we partner again with an OBGYN, an gyn-onc, Alexander Burnett, here at QAMS, and we enrolled women, non-diabetic women diagnosed with endometrial cancer. And so we persuaded them to wait a month before they, from the time that they were diagnosed to be enrolled in the study. And so we gave them metformin as in a clinical trial, a control, which is a placebo, as well as metformin, and this is the treatment regimen for them. And then Dr. Burnett did a hysterectomy of 28 to 32 days after the initial diagnosis, during which time they were either given placebo or metformin, and then he excised the tumors, and we analyzed those tumors. So what did we find? We found that metformin promotes anti-tumor biomarkers in this human endometrial cancer cells. And so these are some of the immunohistochemistry. Immunohistochemistry is wherein we had antibodies to these proteins, and we just incubate these tissue slices on these tumors with these proteins, and they turn brown when they are positive for this protein. And by measuring the intensity of these color reactions, we were able to show that metformin, in the presence of metformin, the tumors have decreased expression of estrogen receptor, which is good because estrogen, if you have less estrogen receptor, you have less binding available protein to bind to estrogens, which is good because lower that would be any lower activity of estrogen receptor, anti-proliferative. We also show that those tumors that are of women who had metformin have increased progesterone receptor. Again, this is good because progesterone receptor is against growth. It's anti-proliferative. We also show that there's increase in KLF9, and this is proof to us that KLF9 is indeed associated with anti-tumor and positively associated with a decrease in in abnormal growth. And there was also an increase in a tumor suppressor PTEN, a well-known tumor suppressor PTEN. So metformin can promote the expression of anti-tumor biomarkers, which tell us then that metformin can inhibit growth of these tumors. Now, because these are taken, women take this, we don't know if this was a direct effect or an indirect effect. So what we did was we took human endometrial carcinoma cells, Ishikawa, and we treated them with metformin or with just the buffer, and showed that there is definitely an inhibition of proliferation of this Ishikawa cells, telling us that metformin have a direct effect on endometrial cells, on endometrial carcinoma cells. And when we look at some of this some of this expression of those genes that are affected by metformin, we did see an increase in a decrease in estrogen receptor alpha, an increase in KLF9, an increase in progesterone receptor and progesterone receptor B, and an increase in PTEN. So again, this tells us that indeed metformin can work directly on endometrial cancer cells and therefore could be a possible agent for decreasing tumor growth. And so this is something that that has been, how would you say, replicated by other laboratories, that metformin can indeed be, decrease, can be an anti-cancer agent. And many clinical trials now, I think one in MD Anderson and several other sites are doing this experiments where they're administering metformin to some of this endometrial cancer patients. We, however, are not able to continue these studies because it's very difficult to get how would you say, patients to be enrolled in the program. It's a small program here at University of Arkansas, as opposed to the MD Anderson and other bigger hospitals. But we were able to, make one of the first demonstration that indeed metformin might be a potential agent against endometrial cancer. So we, for this studies in our clinical trial with Dr. Burnett, we had selected patients who are non-diabetic because we did not want to confound the response to metformin with diabetes and hyperinsulinemia and high glucose. So we did a study where we asked, is metformin effective under diabetic conditions? And this is the project that was, started in collaboration with Dr. Michael Velarde and his student, Sofia Saguyod, when I was Balik scientist at the UP in 2017. And again, as Dr. Giselle has mentioned, this was a project that she started with the UP office of the vice president and in collaboration with the OST And so Sophia was an awardee of the grant to come to my laboratory here at UAMS and ask this question: is metformin then effective under diabetic conditions? So for women who have diabetes, and could you impose metformin on them? Unfortunately, we don't have the patients. We didn't have the patients to do these experiments. As I've mentioned, it's very difficult to enroll patients in our hospital, so we could not expand the clinical trial. So what we did, the next best thing we did, is to have an in vitro study where, again, we had these human Ishikawa cells. These are cancer cells, endometrial cancer cells, and we exposed them to normal levels of glucose and high levels of glucose. And we measured their proliferation as well as expression of some genes that are associated with tumorigenesis. And so what we found is this. The answer is no. If you have an superimpose of cancer and diabetes, metformin is not very effective. And if you just look at this, CCND1 is a cyclin D1. It's a marker for abnormal growth. Higher levels of CCND1 means higher proliferation. And you can see from here, if you just look at this, that metformin can decrease the proliferation, the expression of cyclin D1, therefore a decreased proliferation. But in the presence of high glucose, you don't really have a significant effect. And looking at the expression of progesterone receptor, you can see that under normal glucose, metformin can increase progesterone receptor B. Progesterone receptor, as I've mentioned before, is a mediator of progesterone action, but in the presence of high glucose, you don't see that effect. So there is a glucose-dependent effects of metformin on the PGRB isoform expression. And, women with endometrial cancer, they are given a progestin therapy, but they become resistant after a long time to progestin therapy. And so if you superimpose that most of these women are diabetic, that it is not, that's why it is difficult to treat endometrial cancer for women who are who is diabetic and with endometrial cancer, as because that the high levels of glucose, hyperinsulinemia, hyperglycemia can negate the effects of metformin, and which means then that it's probably best to control diabetes in these patients. So before they're given a progestin, so that then they'll be more responsive to progesterone therapy. So all the studies that I have told you suggests or supports the concept that prevention, yes, we can prevent. We may be able to prevent some of these pathologies in women, but there's no treatment. We're not there yet in terms of treatment. So in terms of interventions to end obesity, diet, physical activity, reduced exposure to environmental carcinogens are all preventative effects to inhibit for this pathologies in women, because all of this can cause inflammation, all of this can cause increase in insulin and production of growth factors, and which can drive abnormal growth of these tissues. We have found that metformin under certain contexts can be good for endometrial cancer, but it cannot be, but if a woman is diabetic, it might be more difficult to address or to treat that to treat endometrial cancer. And so progestin therapy, which is a normal course for endometrial cancers, is not as effective in diabetic women. We also have, our studies also suggest that anti-inflammatory agents, anti-estrogens, progestins, those which are antagonists, which acts against certain proteins that promote growth, could be potential candidates for treatment, but it cannot be one shot. It has to be a combinatorial therapy to address these pathologies. And what we don't know is that how do we deliver these drugs? Local versus systemic. It's very difficult to do local right now. How do you administer, a drug directly to the uterus? It's not possible at this time. But and systemic can have, if you just inject them, it can cause other effects because there are other organs that might be affected by these drugs. For example, the breast could be very sensitive to progesterone, and you could inhibit the action of, or the ovary can be also responsive to the steroid hormone. So systemic administration of some of these drugs have serious side effects. So we are not there yet in terms of addressing drug delivery. So this is something, I don't know if there are a lot of younger scientists listening to this talk, but what did I learn from my 35 years having an independent laboratory and working and trying to understand women health issues? I think what I've learned is that research is a long distance marathon. Dr. Yuzo Yanigamachi from the University of Hawaii, who worked till he was 85, has his own lab, and he just retired. He's 92 now. Said that it's just something that you just pursue, slowly trying to find out, putting pieces together to solve a complex problem. Dr. Robert Lefkowitz, who's a Nobel laureate, has said that you have opportunities, however, as a young scientist, to start writing your own stories. You can have discoveries, but it's a long haul. And indeed, we found it themselves. We have, what we have done is based on other people's work. We try to understand what they have found, and we try to, progress it a little bit, and in the process, start, discover a few other things that could help understand addressing women's issues. One thing that I have learned from all of this is that there is unquestionable value of collaborative work. You can't do the process by yourself. I remember when I was working on KLF9, I wanted to do to get the KLF9 knockout mice. The only one who has it is this older scientist from Japan. And no matter how much I wrote him, he would not answer me. And then one time I had a friend and I was discussing with him how difficult it is for this guy to respond, how to make him respond to me. And he told me, well, Japanese guys don't like to answer, don't like to respond to women requests. I don't know if that's true. Anyway, he wrote the guy on my behalf, asking for the mice, and soon enough, the mice came from Japan, and I was able to do most of the work that I had accomplished working using the KLM knockout mice. So definitely in science, you need to collaborate with people because you can't answer the things yourself. And also, the main goal is pipette to patient. Whatever science that we have, knowledge that we obtain, our main purpose is to translate them into new strategies, interventions for improving quality of life. And so the purpose, in fact, that I hope that what we have understood at the moment from work in my lab is that somebody would continue it and try to understand other things that are still unknowns regarding this women pathology. And I would like to thank the mentors in science. This is Dr. Luli Cruz. She was my first mentor in terms of the lab bench. As I've mentioned before, after I finished my undergrad in UP in 1973, I joined her lab. Told her I wanted to be part of her lab, do my master's with her. So I was in the beginning trying to isolate proteins from the conus. We're doing the chromatography. We have this fraction collector, and we'll go through the night and really in determining the concentrations. And so that was my very first beginnings in terms of working in the laboratory. But then I had the opportunity to go to the University of Hawaii as an East West Center scholar, and Dr. Fred Greenwood was my mentor, and he actually opened my view on women's health, because she was working with the uterus using rat models. And then after I finished my PhD with him, I joined Dr. Tony Means' lab as a postdoc, and he taught me in his lab. I learned about molecular biology and cell biology. I should also point out that Dr. Luli Cruz, was not only my initial mentor, he also helped mentor my daughter. My daughter was doing her honors thesis and on the ATAS in Zambales, in Bataan, and Dr. Luli Cruz and Angela worked with Dr. Luli Cruz on that project. And so the many acknowledgements that I have here, members of my laboratory through the years. Dr. Frank Simon is here. He has been my collaborator in a number of studies. We have different projects. His major focus is actually the GI cancer, but we have worked together in a number of different studies related to the uterus and to endometrial cancer. I have all my collaborators here listed in the last 10 years or so. I was fortunate to have funding from many different, funding agencies. And really the non-fun part of doing science here in the United States is trying to get money. Is really a very difficult challenge, but we were able to get funding from all these agencies. And in fact, one time I was funded by the Fashion Footwear of New York. So you try to get your funding wherever you can in terms of trying to put progress at my work. And so the Fashion Footwear of New York funded some of my breast cancer studies. And here I'd like to introduce you to my 5 grandchildren, 2 from my one daughter and 3 boys from my other daughter. So with this, I'm going to end my talk, and any questions is welcome.
Thank you very much for that, Rosalie. That was a very interesting presentation. So I encourage and invite everyone to please type in any questions you might have, or you may raise your hand if you have something to ask. By the way, Dr. Luli Cruz is actually here in the audience.
Oh, Dr.
Yeah, she's
Cruz,
here.
hi.
Okay, I do have a couple of questions from the audience. The first one comes from Aimee Del Mundo. Is it a decreased level of KLF9 a result of loss of transcription? If not, what could be the cause?
Yeah, KLF9 could be. It could be. We have found that factors could decrease KLF9. So, in fact, at the level of RNA. So I think then that it's due to a decrease in transcription. So, but it's never a real decrease. It's not 100, like a real decrease. So I think that in terms of endometrial cancer, for example, where there is a decrease, it's a contributor, but it's not the sole, the sole reason for endometrial cancers. So there are many progesterone receptor, for example, or estrogen receptor. There's a lot of other co-regulatory proteins. I think KLF9 is one of them. So under certain contexts, I think the KLF9 loss is involved in these pathologies. But I think it is mostly correlated with inflammation. KLF9 has now been associated with inflammatory process.
Okay, thank you for that. The next question comes from somebody who's watching on in YouTube. It's Dr. Angeline Lau. Is it possible that higher dosage of metformin is needed in order to target both endometrial cancer and diabetes?
I think it is possible, but there's a lot of side effects
Mm-hmm.
with metformin. You know, the kidney, there's a kidney lactoacidosis is associated with high levels of metformin. So possibly, but I think that it may, one has to watch that very carefully. I wanted to also point out that it's very, there are a number of laboratories have tried to show that metformin affects a proliferation of Ishikawa cells. So there were many studies prior to number of studies prior to us, but they were adding millimolar concentrations of metformin on cells. Well, physiologically, you never get millimolar concentrations of metformin. A cell in vivo is never exposed to millimolar concentrations. In fact, when you take in metformin, the serum levels only goes to about 40 to 50 micromolar. So in our studies, we went a little bit to 100 micromolar, but we never get into the millimolar. It just becomes pharmacological. So any any results that you go for that you use millimolar concentrations are really, I don't think irrelevant. So that's why I think in some of the studies you always have to make sure that the doses you add to the cells, if you're doing in vitro studies, has to be correlated with
Mm-hmm.
is what is the systemic levels of those agents.
Okay, I'm not so familiar with the level of metformin needed for people with diabetes, but how does that compare? Like for the tests that you conducted, you mentioned that these were in the micromolar levels. Those levels, how does it compare to the typical dosage given to patients with diabetes?
Yeah. So what we have we administered in this clinical trial that we did, small clinical trial, we consider it a
Mhm.
pilot study, is the same dose that
Okay.
the diabetic women get. So it is, but they are non-diabetic.
Okay.
So so we gave them the same dose of metformin that diabetic women, but these are non-diabetic patients. So I the problem with metformin, there are studies now, is that with endometrial cancer, it's never. Sometimes it works, sometimes it doesn't work
Mhm.
with diabetic patients, and I think it's because there. I don't know. Just maybe the serum levels of glucose are just really interfering the effects of the drug.
I see. Okay, thanks for that. Okay, maybe I have another question with regards to KLF9. So you mentioned that there is some correlation with KLF9. Do you see, I mean, sometime in the future, do you see any possible directions on how future work can possibly, either manipulate or control KLF9 to, you know,
Yeah. So what we have to do is to find out what really regulates it. I think glucocorticoid regulates it. There's studies there coming out. In fact, there's a lot of work coming out of China on KLF9. It's really, and then also here, in terms of liver, Frank here is working on KLF9 in GI tract cancer,
Mhm.
colon cancer. So there is a lot, but you have to understand what is the context for it. And I've never really gotten to what really controls it and why does it get lost. In some of our studies, most of our studies, we use really an artificial system in order to understand its function. We have the knockout mice, and also we use siRNA, small interfering RNAs, to decrease the concentration of KLF9 in cells, so we could see a response. We don't know. I'm not going after 35 years in the lab, I don't think I will spend another whatever. So
Yeah,
I
and
think
hopefully we can have
into the
young scientists. Yeah, next generation.
yeah, that's right. Maybe the next generation.
Thanks for that. Are there any other questions from our audience? Giselle, is there something you'd
Ah,
like to?
yes. I'd like to congratulate Rosalie. Very relevant to all of us
Mhm,
for young mothers who are, say, suffering from gestational diabetes as well
mhm,
as those
mhm.
in menopause. I'm just curious whether there is a link between the regulation via the epigenetic enzymes, HDACs, for example, and the hormonal and KLF9 regulation. Have there been studies on this? Because you talked about, well, diet of the pregnant mothers, and that's a good example of epigenetic
Epigenetics. Yes,
regulation.
yes,
Yes,
definitely. Yeah,
yeah.
definitely. It's an epigenetic mechanism. Again, I have not gone through that. A number of people have starting to do that. It's very complex because in terms of the epigenetics, there are many other hormones, enzymes that are involved. Um, so definitely, that's what where it's going. And in terms of KLF9, probably is there has not been any report at this point. Uh, they've found that KLF9 can be ubiquinated. A ubiquitin component can be attached to the protein, and therefore it can be degraded. So it can be also not just at the level of transcription, but it also at the level of protein stability. So that then so it's a complex thing that are trying to be that other laboratories are working on. There's KLF9 now. If you open if you open the PubMed, there's so many work on it now in terms of in terms of endometrial, in terms of cancer in general, not just endometrial cancer. But yeah, so epigenetics definitely, and in terms of this transgenerational, maternal diet affecting the progeny, it's definitely epigenetic. Yeah.
Thanks,
Mhm.
Rosalie. I think for the benefit of our audience, those who are not in this field of DNA regulation, so the HDACs are histone deacetylases. So
Mhm.
histone regulation, the proteins that are intertwined with the DNA regulation of that is also important, right? For yeah,
Yeah, that's right. Yeah, it's a
I,
very complex process.
very
I don't go into it.
yeah.
Yeah.
So thank you so much, Rosalie.
Thank you,
Thank you.
Giselle. Seville, did you want to say something?
Like to say something?
Yep.
Hey, Rosalie. Rosalie Seville.
Oh, hi. Hi, Seville. How come you're not showing yourself? I cannot see you.
Myself.
Oh, because you're sharing the screen, but I think Seville has her camera on.
Ah, do you think I should close this so that
You
then
may,
I can
if you want. Yeah, yeah, I can see the others. Yeah.
do that? Stop share.
Yep.
Yeah,
There
nice.
you go.
How are you? Your hair has grown.
Well, there's Giselle too. And there's your hubby.
Oh, I see Giselle. I saw Giselle. I didn't see Dr. Albano. Yeah,
Oh,
I
oh,
guess I
Dr.
did.
Lou is here as well,
She's
yeah.
gone.
Thank
Oh,
you.
yeah.
and Dr.
Yeah.
Lily Cruz. Nice to
Luli.
have everyone here. Good morning.
Hi, Luli. There's Luli. And as Luli
Yeah,
said,
hi Rosalie.
Hi. Yeah, my first mentor. Yeah. I always go to Luli for help.
Oh, Rosie, Sophia is watching as well, by the way.
Oh, I
Sophia,
can see Sophia. Hi, Sophia.
Hi, Dr.
Oh yeah,
Lucy. Great
yeah, yeah,
talk.
Hello.
yeah. It was a
Yeah,
really
Hi,
Sophia
interesting.
worked,
Hi, Dr. Frank.
Sophia worked very hard in the lab. Yeah, she only had 6 months to do her thesis and she
Yeah.
finished it.
Thanks
Wow,
for the picture,
congrats.
Dr. Lucy.
All right.
Oh,
So, are there any other questions from our audience?
I want to congratulate you. for a beautiful research life.
Yeah.
And
Oh,
can you
oh,
hear me?
there's Lou.
Yes,
Hi.
yes, Will. Yes, we can hear.
Oh, I can be heard.
Yes.
Okay. And this is a product of persistence, patience, diligence, and everything else, wrapped into it.
Well,
Um,
I think I think everybody knows that we all got help somewhere, you know.
yeah, right.
Yeah,
Yeah.
yeah. So,
Nutrition is so, um, important. But I have a
yeah.
question for you.
Okay.
Mhm.
You've been in this field for a long time,
I know. I
studying
know.
all these different, female diseases.
Yeah.
Is there a commonality? Is there some sort of a pathway that can tie up all these different diseases?
Yeah, I think the thing is the estrogen progesterone signaling. That's it. That's it. And so any dysregulation of that can can cause they're very potent. They're very potent hormones, and so but so as I mentioned before, there are so many ways where that could be dysregulated. Receptors, steroid hormone synthesis, but it's really estrogens. The estrogens are the culprit. So the thing is that, and they're affected. Their synthesis is affected by many factors. Obesity, adipocytes, the fat cells make a lot of estrogens. Uh, so so the control of estrogen, some of those environmental exposures, they have estrogenic activities. Some of those estrogen compound, I mean, not estrogen, the environmental um, agents that are, they have plastics contain estrogenic compounds, a lot of things. So I think that it is, it has to be, our exposure should be decreased to that. Um, yeah.
Okay, I have a question. I'm not so familiar with the current treatment, for example, in cancer. So, what's the most common treatment being done now to address cancer in women in particular?
I think the first line of defense is progestin.
Mm-hmm.
Yeah, but then they become resistant to progestin because
Okay.
progestin can downregulate its own receptor. So when there's no progesterone receptor, the protein is diminished. Even if you keep on giving progesterone, there's no mediator of that action. So um, I think that's one thing. I can think that's so. But the thing is that you cannot really. You can only treat it, but you cannot shrink the tumor after some time, so you have to remove it. That's the thing, is to remove. So early detection is good, because then it doesn't grow, um, and
Mm.
but when it's metastasized is when it can be deadly. So early detection is good. Mhm.
Okay, we have another question from Dr. Sean Raimundo. This may be out of topic, but can you comment if there are possible effects of the COVID vaccinations on fertility and pregnancy?
That is very controversial right now. I was actually listening to a webinar yesterday from the people in England, how they can affect menstruation. And of course, if menstruation is a measure of cyclicity and fertility, and they're not sure yet. So they don't know. I think some studies do say, but it's all small studies, and so they
No
they they
conclusions.
don't know yet. I think there's a lot of things that's not known about COVID, but they know that COVID, if you take the vaccine, that the vaccine can be transmitted through the milk, and therefore can your child can be protective if you are breastfeeding. So it's for mothers who, who wants to protect their child. Again, it's it's something that that can be, that, again, prevention is best rather than treatment at this point, if we can prevent it. Yeah.
Okay, Rosalie, may
Sure.
I? So, Luli knows my daughters, both of them. One of them is an MD.
Yeah,
She's
I know. You told me. Uh-huh.
and
Yeah.
Luli, she just gave birth to her second son, and when she got pregnant, she had COVID. So she had
Wow.
COVID, yes. But then there was no. It's all okay, because fetuses and babies are, as we know, do not have a high level of expression or no expression of the ACE2 receptor. Okay,
That's
now
right.
after she got the COVID, she became part of a clinical trial to measure antibodies. So I don't know the result, but then
Mm-hmm.
now she also becomes maybe a part of a study for her baby to have the antibodies to COVID passed
Very
on
good.
to her
Mhm.
son. Okay, so
Very good.
that's my first comment. My
Yeah.
second, Rosalie, is this. So there was a very active PAASE member. She passed away already from UST, and she's Gloria Bernas. Bernas. Everyone
Oh,
knows her from way back. And she had this study on genistein from soya, and it's how it's related to estrogen receptor activity, because we know that it's like an analog, or it's like similar in structure to estrogen. So it's a steroid as well, or it's a isoflavone. And
yeah.
so we would like to know, because you are also studying the diet and nutrition that might help prevent, or maybe be a factor to the development of women's types of cancer. My third question or comment would be about insulin itself. Has there been a study on just getting the insulin shots at low doses to control diabetes, rather than just giving metformin? So those are my comments.
Yeah.
Questions.
Yeah. So in terms of your daughter, that's scary. But what the studies have been showing is that placenta, that it doesn't cross the placenta. So which is good, and so then the baby is protected from COVID, because of the placenta has very little. They have some ACE receptors, but not as much as the lung and the nasal, and so the baby, that's why the baby was it's good, it's again something in nature that protects the baby. In terms of genistein, I actually have done some studies on genistein itself. It's estrogenic under some context. So what we have found is that if you give estrogen to older women, they can be actually they have a negative effect in terms of breast cancer. It's best to give them when they are young, then they are protective. So somehow, again, this is a context. We call them a window where they have a most positive effect. But after that window, it now has a negative effect. And again, maybe it is something to do with the physiology. There, a lot of things happen between young and when you're older, physiology changes, and so that's what it is. And in terms of insulin, and in terms of metformin, insulin is a growth factor. And so then if you give it systemically, it might not go to the right target. So that, there's a lot of insulin receptors everywhere. And so maybe it can go to a different target and may have a negative effect on that. So again, the question here is, how can you maybe administer it locally to the target that you want to, rather than systemically, where it goes through the, through this bloodstream and can go anywhere in other parts of the body. Yeah. So yeah.
Thank
I
you.
hope our medical doctors, if there are medical doctors here, would be, I'm not a medical doctor, but I think that is probably, that's what I think that's one of the, they have this now, the nanoparticles that can go and target the stuff, those who are doing engineering work as a, would be, that's a challenge for them to those doing nanoparticles, I think. I
Thank
had written a grant one time because we found that Notch induces endometriosis. So I was suggesting that if we have Notch antagonists, which are present, those components that would be against the Notch could be used to do to inhibit endometriosis, and in some cases they're being used for cancer. But I didn't get the grant because the question was, how do you know it's going to the right spot? If you inject this, you got to go through, like, deliver it directly to the tissue to be able to control it. And I did not have an answer for that. So
Okay, thank
Uh,
you very much for that. Yes, was somebody?
yeah.
Yeah.
Rosalie, next time you come to NIH,
yeah, I hope I can. Yeah. Thank you.
yeah.
Yeah.
Good luck and congratulations again.
Okay,
Good
thank
luck, and
you.
thank you all for coming. Thank
Thank
you
you.
all.
Okay, but before we go, I just wanted to plug next meetings, next Friday's webinar. Okay, let me just share my screen. Okay, hold on. Okay. So next week's webinar is actually organized by REC4, which is on agriculture, aquaculture, farming, and food. So aquaculture challenges and opportunities through the COVID-19 disruption and beyond. So this has been organized by Dr. Menchi Lagman, and the speakers for next week are Meryl Williams, who is in Gender in Aquaculture and Fisheries Asia Pacific Fish Watch, and Philip Ong, the president of Santa Feeds Corporation. So we will start at 8:30 in the morning on May 28th. So for those of you who are interested to join, please register at bit.ly/rec4 _ May 28, or you can scan the QR code, and it will take you right to the registration portal. And again, please, you can watch our webinars in our YouTube channel, PAASE Webinars, and of course, all data and information on our activities can be found in the website of PAASE, which is at paase.org. Okay, so I guess
Thank
that's it for me. Thank you so much,
Thank you very
everyone.
much.
Have a good
Thank you
day
for this
ahead.
opportunity. I
Good
enjoyed it.
evening.
Yeah.
Good evening
Bye
for
bye.
those of you in
Bye,
the US.
Lulu.
Bye.
Hi.